Hello everyone and welcome to Molecura. Today we're going to be talking about mechanism of action of different antifungals and how they are going to help treat fungal infections. Treating fungal infections are usually going to be challenging, and the difference between fungi and bacteria lies that the bacteria are prokaryotes, so it's going to be more easy to find medications, antibiotics that are going to be targeting bacteria because their cell structures are fundamentally different from that of humans. But fungi are eukaryotic cells like us and like the mammals. That's why treating fungal infections is going to be challenging because they have very similar cellular structures to us. Now there are different components in the cell wall of the fungi that we can potentially target and treat. In this picture that we are having here, we are going to have a picture of a cell wall. This is the black is going to be the cell wall, and the orange structure we have drawn here is going to be our plasma membrane, and this plasma membrane is going to be an important target as well. The main reason is because of the plasma membrane is made of ergosterol, and that is in contrast to us, to human cells, which are made of cholesterol. So that is going to be an important target for antifungal medications. The cell wall is going to be made primarily of structure called chitin. So fungal cells in their cell wall are going to have polymers of chitin, and that is in contrast to bacteria, which, if you remember it, is going to be a polymer of peptidoglycan. Also, polysaccharides that are there in the cell wall of fungal cells are going to be polymers of glucans, and that is in contrast to the bacteria, which we are mainly having N-acetylglucosamine, or we are having N-acetylmuramic acid, which we're going to talk about those in a separate video. So both of these are going to be a legitimate target for antifungal therapy. So the first antifungal that we're going to talk about are those who are going to bind to ergosterol, which is there in cell membrane, and create some pores in it. So we have antifungal medications that are going to be creating some pores in the cell membrane. So there are two medications that can do that. One of them is called Amphotericin B, and the second one is called nystatin. These are the two medications that can create this kind of pores in the cell membrane of the fungal cell by binding to ergosterol. What happens when this pore is created is that many of the potassium and the electrolytes that are there inside the cell are going to leak out, and that is going to cause cell death. So this is a process by which the fungal cell is going to be killed by nystatin or by Amphotericin B. These medications are going to bind to ergosterol on the plasma membrane and are going to create pores in it, which is going to leak the electrolytes outside the cell and is going to cause the death of the fungal cell. Now, the second antifungal that we're going to be talking about is an antifungal that is called Caspofungin. Caspofungin is going to be targeting the cell wall of the fungi. Here we have our Caspofungin. Caspofungin is going to be targeting the cell wall. To understand the function of Caspofungin better, we are going to have an illustration here drawing a structure of the fungal cell wall. So this is going to be our plasma membrane, the plasma membrane of fungi. This is the plasma membrane. This is going to be made of ergosterol, and on top of that we have a layer of chitin. So this is going to be our chitin. So this is going to be our chitin, which is going to be situated right on top of the plasma membrane. This layer is going to be called chitin, and on top of this we are going to have a layer which is mainly known as the glucan layer. So the glucan layer is going to be situated on this. And on top of that, we're going to have manoproteins, which are located on top of the glucan layer. So one of the enzymes that are responsible for making this glucan layer in the cell wall is going to be inhibited by caspofungin. And this enzyme is going to be called, let's try to write the enzyme here. So the enzyme that is getting inhibited by caspofungin is actually going to be called beta 1,3-D-glucan synthase. So inhibiting this enzyme is going to inhibit the formation of glucan layer inside the fungal cell wall. This will result in defective fungal cell wall, and it will result in osmotic damage to this fungal cell, and it will cause death of the cell. The next antifungal that we're going to be talking about is something that is going to disrupt DNA synthesis inside the cell. So when nucleotides are getting synthesized inside the fungal cell, we are going to have conversion of uracil, which is a pyrimidine, to thymine, which is another pyrimidine. The enzyme responsible for this reaction is called thymidylate synthase. And the inhibition of this enzyme is going to inhibit DNA replication. But how does this happen? We are going to have here a medication that we call flucytosine. And for flucytosine to get inside the cell, it's going to need a specific enzyme that is only going to be available with the fungus. So this flucytosine is going to get inside the cell, and to do that, it's going to need the help of that particular enzyme. One thing we can do is to add Amphotericin B to our treatment with flucytosine. And what does this do is creating that pore that we talked about at the beginning of this video. And this pore is going to be created on the fungal cell wall, and it's going to be allowing the flucytosine to enter the cell more easily to have its therapeutic effect. So here, for flucytosine to get inside the cell, it could use the help of Amphotericin B to do so. So Amphotericin B could have synergistic effect when it comes to the action of flucytosine. That's why, for example, in treatment of cryptococcal meningitis, we're going to give flucytosine and amphotericin together. Now, what happens when flucytosine gets inside the cell? Our flucytosine is going to be further converted into another molecule that we call 5-fluorouracil. And our 5-fluorouracil inside the fungal cell is going to be inhibiting the thymidylate synthase. And this is going to result in decreased DNA synthesis inside the fungal cell. So the next antifungal that we're going to talk about is going to be one that is going to be inhibiting mitosis inside the fungal cell. So what we have drawn here is actually a nucleus inside the fungal cell, which has the chromosomes lining in between for mitosis to happen. So what happens here is that we are going to have our mitosis. There are going to be microtubules attached to the chromosomes while mitosis is happening. And what happens at the end of the mitosis is for these spindles to go backwards and to separate these chromosomes from each other. We have an antifungal that is going to inhibit the polymerization of these microtubules, and this antifungal is called Griseofulvin. So Griseofulvin is going to be inhibiting the microtubule formation in mitosis, and it's going to inhibit the fungal cell from growing and multiplying. So this is our fourth group of antifungals that we are going to talk about. We will talk about two more groups of antifungals, and those are medications that are going to inhibit the synthesis of ergosterol in the plasma membrane, draw the short pathway of ergosterol synthesis in the fungal cell. So this pathway is going to be started by a molecule that we call squalene. So squalene is going to be our starting point in this pathway. So the first reaction is our squalene getting converted into a molecule called squalene epoxide. And this is happening through the action of an enzyme that we call squalene epoxidase. And our squalene epoxidase is going to be the first enzyme that is getting inhibited in this reaction. And the inhibition of this enzyme, it's happening through a medication that we call terbinafine. So terbinafine is the first medicine that we are talking about in ergosterol synthesis. So our terbinafine is the first one that we are talking about here. The next structure is going to be lanestrol. We are going to skip the other parts of this pathway for the sake of simplicity. And our lanestrol is going to ultimately be converted into ergosterol, which is the final product of our pathway. Here we are going to have our ergosterol. And this ergosterol is going to be implemented inside the cell membrane. So there is another enzyme over here that is going to be called lanestrol 14 alpha D methylase. 14 alpha D methylase is one of the enzymes that is going to be working on converting lanestrol into ergosterol. There are more reactions happening here, but again, for the sake of simplicity of the diagram, we are going to show it like this. So our final class of medication are going to be class of antifungals that are going to inhibit this lanestrol 14 alpha D methylase. And this is going to be the class of medication that we call as azole antifungals. So azole antifungals are going to be our next class of medications here, and this is going to be medications such as ketoconazole, fluconazole, itraconazole, or voriconazole. So by this we have talked about the major classes of antifungals. Just to wrap up the video, we're going to be going through each one of them very quickly as a recap. So the first one we talked about is amphotericin B and nystatin. Amphotericin B is going to be creating pores in the cell membrane of the fungal cell. These pores are going to cause leakage of electrolytes like potassium outside the cell, and this will cause cell death. The second one that we talked about is caspofungin. Caspofungin is going to actually inhibit an enzyme in the synthesis of the cell wall of the fungal cell. This enzyme is called beta-1,3-D glucan synthase, and this enzyme is responsible in creating the glucan layer of the cell wall. We said that the cell wall of the fungi are made of chitin, which is the main polymer. We're going to have a glucan layer, and we're going to have manoproteins. Apart from this, we also talked about flucytosine. Flucytosine was a nucleoside analog which was getting inside the fungal cell, and once inside the fungal cell, it was getting converted into 5-fluorouracil, which was inhibiting an enzyme that we call thymidylate synthase. This enzyme is responsible for converting the pyrimidine uracil to thymine. By this, it's inhibiting DNA and RNA synthesis and is going to have detrimental effect on replication of the fungal cell. We also talked about how amphotericin B can create a pore that is going to make it easy for flucytosine to get inside the cell. Also, we talked about Griseofulvin, which is another antifungal that can inhibit microtubule polymerization and inhibits mitosis inside the fungal cell. The last two that we talked about are terbinafine, which are class of medications that we usually use, for example, for onychomycosis, the fungal infection of toenails. What happens with terbinafine is that it's going to inhibit an enzyme that is called squalene epoxidase. When squalene epoxidase is inhibited, we are going to have a decrease in the downstream product of this pathway, which is ergosterol, which will disrupt the cell membrane of the fungal. Also, by inhibiting this enzyme, we're going to have an increase in squalene accumulation inside the cell, which is going to be toxic to the fungal cell. In the later stages, we are going to have a molecule of lanesterol, which is going to get converted ultimately to ergosterol. There is an enzyme in between that's called lanisterol 14 alpha demethylase. This is the enzyme that is going to be inhibited by azole antifungals such as ketoconazole, such as fluconazole or itraconazole. By this, we're going to finish our video on mechanism of actions of common antifungals. If you find this type of content helpful, make sure to subscribe to our channel, like the video, and leave your comments to be able to explore more videos on Molecura. Thank you.